Thank you very much, and I'm delighted to be here in the Holy Grail of the French Academy of Sciences. I already spoke once or twice in the Sorbonne with the sealing and the French Revolution, so it's a great honor for me. Actually, I will tell you today about hematopoietic stem cells and host immunity and how they are regulated by light and darkness onset. I will talk about the metabolic regulation, the role of reactive oxygen species, ROS, norepinephrine, TNF, and melatonin. So the blood-forming stem cells have two major roles. You can see on the right-hand side that they need to produce all our mature blood and immune cells on a daily basis. In adults, it's over 1 million cells per second. On the other hand, on the left-hand side, they need to retain the reservoir of undifferentiated stem and progenitor cells. And they do that in their bone marrow niches where they interact with bone-forming stromal stem cells. They are retained in a low metabolic state, low ROS levels, and this way they are chemotherapy resistant. So we are interested in how these two opposite processes are regulated. And I put here the two types of asparagus because both of them are genetically identical. However, as you can see, the green are exposed to daylight, and as a result, they have a more woody taste. While the white, yellowish, they are deprived of daylight. They are grown in the dark, which gives them a unique baby-type, undifferentiated taste. I know we're outside the season, but Paris is well known for the white and yellowish asparagus. So when we look at stem cells in the bone marrow during the day and during the night, you can see that we see two peaks of undifferentiated blood-forming stem cells. One at 11 o'clock in the morning and the other at 11 o'clock at night. And we want to know how are these peaks generated and what are their roles. So in our animal colony, light is initiated at 6 o'clock in the morning and is terminated at 6 o'clock in the evening. And you can see on the left side, 1 hour after light initiation, we have elevated levels of metabolism of reactive oxygen species. The stem cells are transiently ROS high, and at 11 they are again ROS low. And the same thing 1 hour after light termination at 7 o'clock in the evening. Also, there's a transient increase in the metabolism in reactive oxygen species, ROS, which again are downregulated at 11 o'clock. So what initiates this increase in ROS levels? And tumor necrosis factor is one of our candidates. And when we give mice TNF on the left-hand side in A, indeed we see a transient elevation of reactive oxygen species. And when we look in the bone marrow during the day, we see that 1 hour after light initiation, we have a high peak of TNF, and 1 hour after light termination, we have a lower peak in TNF. So this is interesting because TNF is known for inflammatory reactions, autoimmune disease like rheumatoid arthritis, malignancies, but no physiological role actually. And we see one here. So we wanted to know what initiates this TNF secretion. And it's already been shown before us by the group of Paul Frenette in Mount Sinai in New York that light initiation induces secretion of norepinephrine, elevated levels. And indeed, we also see at 7 o'clock in the morning, elevated levels of norepinephrine. Interestingly, we also see a lower peak 1 hour after light termination. So darkness doesn't signal and doesn't have a receptor, but again, we see a peak, a lower peak of norepinephrine. And these peaks are important for the TNF because if we inhibit norepinephrine with beta-2 blockers, we don't see the increase in norepinephrine in TNF. And also if we add the ROS scavenger and acetylcysteine, again, both in the morning and in the evening. So we wanted to know what is the role of TNF, what is the role of norepinephrine. And for that purpose, we took mice which don't have a TNF. They're TNF knockout. There's also an antagonist, and we see that they have abnormal levels, high levels of norepinephrine, and they don't respond in B to norepinephrine signaling properly. If we inject mice with norepinephrine, they will mobilize stem cells into the circulation, but not in TNF knockout, suggesting that TNF is like a traffic light which coordinates a norepinephrine function during the day. So what is the role of these day and night peaks of 7 o'clock in the morning, 7 o'clock in the evening, 11 o'clock in the morning, and 11 o'clock in the evening? So at 11 o'clock in the morning, we see stem and progenitor cells are released into the circulation. And as I told you, we need to generate 1 million cells per second. The cells are released in mice into the circulation, replenishing the blood at 11 o'clock in the morning, but not at 11 o'clock at night. So what is the role of the peak at night? We can also take cells from mice at 11 o'clock in the morning or at 11 o'clock at night. At first, we worked very hard day and night, but then we generated an upside-down room where night occurs during the day. So now we can compare the two, and we can take these cells for transplantation, like in clinical stem cell transplantation of bone marrow transplantation protocols. And we see a huge difference between the cells at 11 o'clock in the morning in the bone marrow and at night. The night cells engraft much, much better and much, much higher levels. But why? If we look at 11 o'clock in the morning and 11 o'clock at night, the levels are pretty similar. What is the reason for the difference? So as you know, the darkness hormone, melatonin, is increased. It is generated in the pineal gland, but there are also some reports it can also be generated in the bone marrow by immune cells, myeloid cells, phagocytes, monocytes, macrophages, and also bone-forming stroma cells. And when we look at melatonin levels, we see, as expected, a very high peak at 11 o'clock at night. And again, if we look at TNF knockout mice, they have eyes, they can see, but they don't have this elevation in melatonin at night. Also, normal mice, if we treat with the TNF inhibitor, they do not elevate their melatonin levels. So again, TNF in the morning and TNF in the evening, like traffic lights, regulates norepinephrine effects and melatonin effects. And to summarize what I've shown you so far, in the morning, stem cells are primed metabolically for their proliferation, differentiation, common myeloid and common lymphoid progenitor cells, and at 11 o'clock, they mature leukocytes egress to the circulation, replenishing the blood with new cells, which all have a finite lifespan. While at night, the reservoir of stem and progenitor cells are regenerated. After all the loss during the day, you need to regenerate the arsenal so that we'll have durable blood production throughout life. And we want to know, how is this regulated? And how do the stem cells at night have much better stem cell phenotypes, homing and repopulation potential, like the yellow versus the green asparagus? So if we look at some of the cell surface markers, and in mice, the stem cells have lymphoid markers which are called SLAM, we see that at 11 o'clock in the morning and at 11 o'clock at night, there is a huge difference. The number of stem cells are similar, but the SLAM markers like CD150 or surface c-KIT change dramatically. And so as a result, we decided to see if this is indeed due to melatonin or it's only in correlation. So we woke up early in the morning before a light initiation at 6 o'clock, and we gave melatonin for 5 hours, turning the day into the night. And we see indeed we have higher levels of these stem cell markers. But we wanted to know function, so we transplanted the cells. 11 o'clock in the morning control versus following melatonin treatment. You see already they have a huge advantage in repopulation. And even if we transplant half the cell dose from mice treated with melatonin, they're as good or even superior to the control. So since mice are nocturnal and we are not, we wanted to know if all this is relevant for humans or only for mice. Already it has been shown in immune deficient mice, which cannot reject human stem and progenitor cells, that in these immune deficient mice, the mature leukocyte released into the circulation is different between the human cells and the mouse cells. They have different clockwork and different signaling cascades. However, as I told you, there are cells in the bone marrow, including bone-forming stromal stem and progenitor cells, which can secrete melatonin, including human bone-forming stromal stem and progenitor cells. This was shown in vitro. So we treated immune-deficient mice, which were repopulated with human stem cells, also for melatonin in the morning, and we saw the same increase, suggesting that what we found is relevant for human stem cells as well, although these results are preliminary. So to summarize what I've shown you so far, we see in the morning metabolic programming by norepinephrine and TNF inducing the cells to migrate and to develop proliferation, differentiation, and release into the circulation to replenish the blood. And this is done over a few hours. And at night, we see the exact opposite. TNF and melatonin induce the stem cells to reduce their metabolism. At night, the cells have lower levels of ROS compared to the morning, and to retain the stem cells in the bone marrow in their undifferentiated form. Again, like the yellow and green asparagus. So switching now to bone stromal stem and progenitor cells, because we know our bones are dynamic. We know about osteoclast-osteoblast interactions, which regulate bone turnover and also regulate host immunity. Here, the osteoclasts, when they dig the leucana, they free the yellow blood-forming stem and progenitor cells into the circulation, and it's like a biological clock which regulates host immunity, because viral, bacterial infections all accelerate bone turnover. And we also know that during aging, in particular women after menopause, our bones become thinner, we become more anemic, and host immunity deteriorates. And we were pleased to see this study from Harvard from the previous millennium, from the early 60s. They looked 1 hour after light initiation, and they saw the highest levels of osteoclast frequency, suggesting that the bone turnover is also regulated by day light and night darkness onset, which regulate both bone turnover as well as host immunity, which is synchronized. And indeed, if we look at the bone-forming stromal stem and progenitor cells, you also see here in blue that at 11 o'clock at night, the two populations of bone-forming stromal stem and progenitor cells are significantly elevated. So to summarize this part, we look at the bone marrow as a prism that during the day induces proliferation, differentiation, new blood and host immunity production, new bone formation, and these are all the rainbow colors on the right, while at left, during the night, they do the exact opposite. They regenerate. It's like an anti-aging effect, both for the blood and for the bone-forming stem and progenitor cells. And so the question is, how does this impact on our host immunity? Here is again another old publication from the previous millennium, from 1960. They gave mice E. coli endotoxin to mimic gram-negative bacterial infection. When they gave the mice this challenge in the afternoon, at 4 o'clock, when there's peak levels of steroids, corticosterone in mice and cortisol in human, almost all the mice died. When they gave the same treatment at midnight, almost all the mice lived, the same exact treatment. So we looked carefully to see what is happening, and we give lipopolysaccharides to mimic bacterial infection. And when we give it during the daylight in the afternoon, and we wait for 20 hours, we see a huge increase in neutrophils in the blood and monocytes, innate immunity to fight the inflammatory response. Surprisingly, when we look at 20 hours post-midnight, the same exact treatment, we see opposite effects. There is no increase, not in neutrophils and not in monocytes. Actually, there is a decrease. And again, if we attenuate ROS signaling by giving the ROS scavenger NAC and acetylcysteine, we can attenuate and rescue this inflammatory response. And We haven't looked, but we think that sepsis patients, which is a life-threatening problem, if they will be kept in the dark, maybe it can impact and help their survival. So our data suggests that ROS inhibition can be utilized to attenuate inflammatory responses. So in mice and also in people, but in the other way around, we have an active site and an inactive period for our host immunity. And as you know, when we travel through time zones, we suffer from jet lag, and our host immunity suffers as well. And this is important not only for normal blood and bone production, but also for pathological situations. The group of Ben Ebert in Harvard has shown that core circadian clock genes, which are downstream to light and darkness, they regulate acute myeloid leukemia, AML. And it is also well known that night shifts increase the risk for breast cancer, especially if there are intensive night shifts, and a lot of night shifts over the years, like in the case of nurses, they have a 32% increase in chances for breast cancer. And in this study where they treated a model of breast cancer in mice, they found that the treatment with chemotherapy was more effective if it was administering during the resting phase of the day when glucocorticoid levels are low, and that gave better results than treatment when there was peak steroid activity in the afternoon. So to summarize this talk, we think that day and night are like, and light and darkness onset are like the Oriental Chinese yin and yang of the bone and blood-forming stem cells for their function and maintenance. And unfortunately, this harmony is disrupted during times of malignancy, or even during aging, because during aging there's a dramatic decline in melatonin production, sex hormones, and fertility, which all lead to clonal hematopoiesis, reduced host immunity, anemia, osteoporosis, and leukemias. And we hope that these preliminary results with light and darkness may shed light on new mechanism of how this programming during the day, metabolic programming and reprogramming during the night actually takes place with the chromatin and which are the epigenetic players, so that hopefully we can harness them for improving or attenuating aging and for a better arsenal to fight malignancies. Thank you for your attention. Merci beaucoup.